Okay, next up we have Liz Shea telling us about measuring marine mollusks.
Millions of mollusks. Hello, everybody. I'm Liz Shea. I'm the curator of mollusks at the newly named Delaware Museum of Nature and Science. And I'm speaking today on behalf of my two colleagues, Larry Van Stone, who's the computer scientist who is doing a lot of the work that you will see me talking about, and Alex Kittle, who's our senior collections manager at the museum. Rudiger in the intro text or Elena on behalf of Rudiger mentioned this survey that was done in 2018 that pulled out information from across all sorts of collections around the US and a little bit beyond, that we have about 100 million mollusk shells sitting in collections. Now, that's an enormous number of mollusk shells. It's really second only to the estimated number of insects in collections. And it averages out to about 1,100 specimens per species. And some species, there's considerably more than that. And that means that we're really well situated, we're well positioned to do ecological studies based on these specimens. And so as part of ESB, you've heard, and as part of DigIn, there's a photography component to it. People who take a lot of pictures of shells, I am not yet one of them, spend a lot of time emphasizing kind of just exactly the right position, making sure they're up, facing up, aperture up. They are using a lot of stacking of images, getting really high quality, excellent clarity, a lot of detail, and with a sense that a lot of this is meant for identification, or it's meant for systematic work. But as you'll notice on the image on my left, yep, your right, that this is really just one specimen out of a lot full of 9 specimens. And there's a lot of post-processing that goes on in all of this. And because we're a collection that is not really begun doing any photography, we're a relatively young museum. We've got an extensive collection, but we haven't got 100 years of photographs to kind of sort back through. We really haven't started taking any photographs. So we're trying to start from a position of thinking about everything that we hear in these conversations today, plus a little bit about how students in particular are using specimens or need to use specimens. So the first characteristic that we're thinking through, this first problem, is how do we maximize the usage of these specimens? And a lot of this inspiration came from my work with a group called BCNet, the Biological Collections in Ecology and Evolution Network. And this is an RCN UBE, I think is technically what we are, run through the National Science Foundation. And the idea is to get students to use the collections digitally during COVID or in person in other times. This image is the start point for us, where we had Widener students working in our collection. And when you're working in a physical collection, you are using every specimen that exists in that box. You're not picking and choosing an exemplar from a lot. And that really has stuck with me in terms of what we want to do in terms of our photography experience. We also need to minimize the handling time, because even though only 3,000 species of mollusks are being treated in the eastern seaboard TCN, there's at least 51,000 different species of marine mollusks. And if we're taking multiple specimens, multiple views of one specimen, is that really giving us what we need? Or do we need to be looking and documenting variations in size, color, distribution, location? So my conveyor belt technology picture is something that I'm really jealous of. Because the thought of being able to just slide things through and take a bazillion pictures of them and not have everything roll off onto the floor is really something to aspire to. The other thing that's animating what we're trying to work through is preparing for the future, because we're starting basically at square zero. It seems to me, and I think you would probably all agree, that the future is going to be a lot more automated, and it's going to be computers drawing bounding boxes around things and extracting information. So this is just one of many different examples of the way in which machines are going to start to use our collections activities. And so we've kind of come to the conclusion that we really want to photograph all of the shells. and we want those shells to be ready to measure. And we're trying to figure out what the concerns are about making those shells ready to measure, and not only minimizing the handling time, but making sure we're not introducing error into research publications anywhere. And so we've got basically 4 sources of measurement error that we have to kind of find our way through. And let me see if I can get this thing to work here. Nope, that's not going to do it. Okay, well, I'm just going to tell you that when we're taking our measurements of these shells, we're going to take 4 measurements when we're doing them in a photograph. The very top of the shell, the very widest part on the right, the bottom of the shell, and then the very widest part on the left. And then we have to account for these different sources of error. So the first source of error that we're going to deal with is the fact that shells tilt, right? And that seems very silly, of course. But when you're taking a picture, and the picture on my right, your left, is a plan view, and you might think that those shells are beautifully positioned and that they're all lying in exactly the right spot for you to take a great measurement. But if you look at them on the side view, you can see that there's a huge impact of the tilt of the shell. Now, the specimen on the bottom that's marked small tilt and small impact is what we've been doing most of our testing on. But the larger the tilt, the larger the impact, the larger the measurement error that you end up with. So the second problem that we have to deal with is the fact that we're working with cameras that are actually relatively close to the specimens, no matter how far away they are. They are closer than they would be if you were working in a perfect world at an infinite distance. And Larry Van Stone, who has done a lot of the development of this, was really trying to convince me to have mirrors put up all around the collection, and to be taking pictures as things ricocheted off of mirrors around the floor to get to this infinite distance. But the idea is that at an infinite distance, the light rays are all parallel. And so the picture that you're taking of the shell and the shell that you might then be measuring is exactly right. There's no kind of shadow. When you have a camera set at a known finite distance, though, because of the way the rays of light are shining not from an infinite distance, there's a little bit of a difference, not a huge difference, but a little difference between what is now on your picture and what the actual distance is. And so we've been calling the measurement that you might take on a picture the naive measurement, and the other measurements, that's the one that has to be kind of corrected for this shadow. So the third problem that we had to deal with is that when you're taking a measurement in real life, you're using maybe calipers, and you're taking a very distinct, pretty easy measurement. Everything lines up kind of nicely. But when you're doing this in a photograph, and you're doing it by taking the very tippy top and the farthest out to the right, and the bottom and the farthest out to the left, there's actually an offset that you end up with. And this offset is changed depending on which specimen you're using and what species you're using. And so we've been calling this the yaw angle. So we have to kind of correct to get all the geometry to work. We have to get the measurements to line up so that you're not overestimating the width of anything. So in real life, when you're taking these measurements, you're automatically kind of correcting for this tilt and the yaw, and there's no distance involved because there's no camera involved. But when you're doing it based off of these 4 measurements, you have to think about how to do these mathematical corrections. And this is where I start to wander way off into territory that I'm unfamiliar with, and that is computer programming and a lot of high math, geometry and trigonometry. But the idea is that because of the way that we can use a program. In this case, Larry's written it in Excel. And it's relatively straightforward and easy to use. In order to solve problem 1, we have to figure out what the tilt of this shell is. And we can do that using geometry. The red dots are the dots that are used for the tilt calculation. We're doing this all based on the fact that the snail is being modeled as if it's a perfect cone. And we know that that's not the case, right? But we have to make some simplifying assumptions. If you look on my left, your right, the plan view and the side view, it is really that alpha angle that we're trying to estimate. Because if we know that alpha angle, then we should know in a perfect world what the actual length of this shell is, even if it's leaning into the screen. And here's where the math gets. This is the side note for the math majors. This was important that Larry to Larry that I articulate this, that if we know the 3 dots, there's a no closed form way to calculate what the alpha is. I don't actually know what that means, but that means that we have to have a computer program to help us with this approximation. And this approximation is what this part of what this computer software does. Oh gosh, the time is ticking by. So the second problem we have to check is to correct for this shadowing event. So we've got two different situations that we kind of played with. A perfect world, the camera at a far distance, and the real world, the camera at the known distance. And we were basically asking ourselves, how much worse is it to have the camera at the known distance? Is it really beyond what we can correct for? Because correcting for problem 2 requires this projective geometry to be used. And that's like loads and loads and loads of programming. But this is also what your augmented reality headsets are doing, apparently. And so let's see if I can get you. So the naive measurement here is following the end of these green arrows, because that's where these rays of lights are kind of faking or making your camera think that the end of the shell is at a different place than it actually is. And so what we're really trying to do with all these corrections is find what that little spot is there and adjust the naive measurement and turn it into really what the known actual length of this specimen is. So how important are these corrections? We went through and did a little bit of testing from the known distance, from the far distance. And what you'll see here is in two sets of measurements, the length and the width. And in all cases, the width measurement is pretty robust. It doesn't need a whole lot of correction. And the correction, when it is applied, brings us into about plus or minus 1/4 of a millimeter in our length, in our width dimension. But the length requires a little bit more massaging, because, and you can imagine that that's primarily because of the tilt. So each of these 3 lines is a number of corrections that are applied. The first one is yaw, the blue is yaw, the orange is yaw and tilt, and the black is yaw, tilt, and perspective. And what you will see and what we've got here is that the impact on length is greatest, width is fairly robust. And even if you take it from far away distance, which is not really super useful for other activities that you might use the pictures for, you're still within about 1/2 millimeter. So the next step for this is to move beyond Larry and Alex and Liz and say, can other researchers get accurate measurements from these kind of spilled shells photos? And so we're trying to do a test with some volunteers, with ourselves. We're looking at 12 lots of Neoterebra shells and doing the measurements, and we're only partially through all of this. So I don't really have a lot to say about it, except that it's ongoing and it's looking relatively robust at the moment. In terms of outcomes and recommendations from this activity, what we have learned is, instead of spending a lot of time handling shells and putting them all in that nice up and down formation, we can spill things out and we can turn them all aperture up. And that's the best way for us to get a consistent with previous work and to get a nice, strong, robust result. The software that we're developing is right now, it's like an Excel software. It's still in development, but it's promising. We're really pleased with how the corrections seem to be able to bring us to within 1/2 millimeter of real life. The one thing that was interesting is that it was because of what we're using for this software, we do need to know the height away from of the camera. We need to know what that is, and we're thinking about ourselves putting that number into the image, or at least adding it to the metadata someplace. A weird little quirk is that the measurements need to be taken in clockwise order, but it doesn't matter if the shells are all lined up or if they're just all spilled out. Errors are minimized when everything's close to the center, and the last bit is use the best and longest focal length lens that you can afford. What we did also learn is that this is not necessarily universally applicable across taxa. So this is step 1 for us. We started with the low-hanging fruit, gastropods with rather straight sides. But there are definitely ways where we're going to have to modify and improve moving forward. Right now, we're also looking at flat bivalves and flat gastropods, things that lie down nice and neat. But we're going to have to deal with like the worm snails and things that have giant spines and other additions. In the future, and I see I'm out of time, we've got other things that we're going to do in the future, including we've got a little shortcut in mind where we can just measure all of the shell apexes for different taxa and see if we can do a little less, a little trigonometry, but a little less elaborate activities to do this. And we're hopeful that all of this is going to make everything machine actionable. So I just want to finish up by saying thank you to the organizers, to iDigBio, to Spinach for having us here. These digitization efforts, I mean, the community that's developed around these has been really useful for us as a institution. And that I wanted to point out that BCNet and its progenitor, or its origin really, Blue, have really got us thinking about all the ways in which people are using our data, and that's really exceptional for us. So I'm at the +3 minute mark, I think, I'm sorry. And I'd be happy to answer questions, or we can move on to next.